Hi, everybody. It is an honor to be here today. As Jennifer said, I am a genetic counselor at UT Health Houston and currently working in our first multi-D SCN2A clinic. So this has been an incredible journey and very happy to be here with you all today. Wanted to put in a plug for the research we're doing. So in room Garden B, we still have a couple slots open for tomorrow if any of you are interested in signing up for a research project. And if we do not have slots available, we're also happy to take your information and contact you later for some research. But I feel like I'm really talking to a room full of experts in this, so forgive me if some of this will be review for a lot of you. But genetics can be complicated at times, as exemplified by the previous talk. But hopefully this can help clarify some things for you all. I do not know how this works. Okay, here we go. But what we know is that we have chromosomes, and chromosomes contain all of our genes, and genes are sort of the book of instructions, and they tell our body how to grow and to develop. And we have two copies of every gene in our body. So if we were to think of DNA of a string of letters, we can think of the SCN2A again gene as being this string of letters, and each person has two copies of it. And sometimes you might hear the word allele been thrown around in genetic testing reports or as you read different papers. And basically, that's just a fancy word to say that it's a different version or a copy of that gene. What we know, and that I'm sure most of you here in this room know this, is that the SCN2A gene encodes a sodium voltage gated channel alpha subunit 2, or sometimes you see it referred to as this 1.2 channel. And we know that this protein is very important because the sodium channels play an essential role in the ability of a neuron, so our brain cells, to communicate with each other. And that's because we know that these sodium channels allow these sodium ions in and out of cell. And this flow of ions in and out of cell is what generates the electric signals that allow our cells to communicate with one another. And what we know is that whenever there are genetic variants or mutations in this gene, this flow of ions can be impaired. So it just is not what we expect. As mentioned before, we know that each person has two copies or alleles of that SCN2A gene, and one copy comes from the mother and the other copy comes from the father. What we also know is that in the vast majority of individuals with SCN2A related disorders, one of those SCN2A alleles has a variant, or sometimes you also hear the word mutation. So here on the slide, we can think of it again, of the gene being that string of letters, and one allele or one copy of the gene would be normal, or what we sometimes call wild type, and the other copy of the gene has that genetic variant or a mutation that then impacts how the sodium channel works. We know that there are many different types of genetic variants that exist, and this is by no means a comprehensive summary. Otherwise, I'd spend the whole talk talking about that. But we know that a couple of different genetic changes we can have are single letter changes. So for example, here in this example, you see that the letter T was changed to the letter C. We could also have insertions, meaning so there will be additional letters in the gene. And on the opposite end, we can also have deletions. So some of those letters could be missing. And certainly the size of these insertions and deletions can be much larger than just a couple letters. What we also know is that depending on the type of genetic variant or genetic mutation, there will be different consequences on the protein level. So what we know is that those letters of DNA code for what we say amino acids, and amino acids are just building blocks of a protein. So again, when we have a change in the DNA level, we have a consequence on the protein level. So a word you probably have heard before is a missense variant. And this is a common type of genetic variant where we have a letter change, often on the DNA side, and then that now changes an amino acid on the mRNA or the protein level. So for example, here we see if the wild type, if that letter A was changed to a letter C, instead of coding for leucine, the amino acid, we would now be coding for phenylalanine, so a different amino acid. And what this could potentially look like in the genetic testing report is that you would see, the strings of letters and numbers that I'm sure all of you have seen before. And a clue that this would be a missense variant is that, as you can see, we have that leu and then the number, which would be the position where that change is happening. And then the amino acid following that is a different one. So that would be a simple change of amino acid caused by a missense variant. On the other hand, you may have also heard about genetic variants that are referred to as protein truncating variants, or PTVs for short. And we call these type of genetic variants whenever a change in the DNA in those letters leads to a shorter protein. So for example, in this case, again, if we were looking at that change in the letter A, if we now change it, or sorry, I think there's a typo there, but if we were to change T to an A, instead of coding for the amino acid leucine, we'd now code for a stop codon, meaning that the rest of that protein will not be there. So we're having a much shorter protein than what we would have expected. And again, if we were to think about this in a genetic testing report, what we often see is that you'll see a star after that position, or sometimes you'll also see the letters TER, and that stands for termination, or also known as a termination or a stop codon. And again, that would just indicate that we have a PTV or protein truncating variant. What we also know is that the different SCN2A genetic variants affect that sodium channel in different ways. And as many of you here know, some of those genetic variants can lead to an increased flow of sodium ions through the channel, which we commonly refer to as gain of function, while other genetic variants lead to a decreased or no flow at all of sodium ions through that channel, which we commonly refer to as loss of function. We also know that, broadly speaking, that loss of function and gain of function are correlated with an individual's phenotype or clinical presentation. So typically, when we see these gain of function variants, we expect a phenotype where we are having epilepsy prior to 3 months, or we can also have the self-limited form of infantile epilepsy. Whereas on the loss of function end of the spectrum, we're often seeing epilepsy happening after 3 months of age, or we see individuals with just autism and ID without any epilepsy. But again, as demonstrated by our previous talk, we know that these correlations are not as simple as what is shown here, and that we're still learning a lot about it. But if we were again to sort of generalize things, we know that broadly speaking, those missense variants, so when we have a single change in amino acid, those can lead to loss of function, gain of function, or a mixed function effect, whereas most protein truncating variants would lead to a loss of function effect. So now just kind of jumping into the next part of the presentation where we're talking about inheritance of SCN2A related disorders. We know that in general, genetic variants can be inherited from either one of our parents. However, in most individuals with SCN2A related disorders, these genetic variants occur spontaneously, or what we also refer to as de novo, meaning that they randomly happened earlier in development and were not inherited from either parent. We also know that these de novo variants either happen in the sperm or the egg, or very soon after fertilization during that zygote stage when the egg and the sperm have just met. And again, in these cases, the variant is expected to be present in every cell of that affected individual. However, again, neither parent is expected to have that variant. And so then the question is, how do you know if your child has a de novo SCN2A genetic variant? So as a rule, it's always recommended, or as a suggestion, it's always recommended that the parents of a child affected with SCN2A related disorders undergo genetic testing so that we can see if they carry that specific variant that we observe in their child. But if we do test both parents and both of their tests come back negative, that's when we would refer as that genetic variant being de novo, or again, we're expecting it to have happened in the child for the first time. So when we think about an individual affected with SCN2A related disorders, if they were to ever have a child, we would expect that each of their child would be at a 50% risk of inheriting that genetic variant in SCN2A and also be affected. However, when we think about the parents of an individual who is affected, we say that the risk for them to have another affected child is very small, but genetics is one of those fields where we never say never, we never say zero. And the reason behind this is because there is something called germline mosaicism. So what is mosaicism? I think this is one of those concepts in genetics that can be confusing. So hopefully this will help clarify it. But mosaicism occurs when a genetic variant occurs later in the development. And again, this can still be in utero in one of those first few stages of cell division, or it can be later. And mosaicism, again, is when that genetic change is present in some of the cells, but not all of them. And typically, the later in development a genetic variant occurs, the fewer cells would be affected. So as you can see here on the first drawing, it happens in that 2-cell stage. So we would expect majority of the body to have that affected variant versus if it happens much later when the organs themselves are developing, then maybe that genetic variant might be restricted to just, for example, just the brain. Why does mosaicism matter at all? We know that there have been reports of individuals with mosaic SCN2A variants reported. From the data we have so far, there doesn't seem to be a significant difference in terms of age of onset or severity for those individuals that have mosaic variants. But again, it's something that needs to be studied more so that we have more data to say this for sure. But what does matter in terms of mosaicism is that the chance of passing SCN2A to future offspring is slightly different. And we'll talk about that next. Again, just to reiterate, when an individual is found to have a mosaic SCN2A variant, neither parent is expected to carry the variant. And again, that's because the genetic variant was not present in the egg or the sperm, but it happened at some point later in development. And how would you know if your child has a mosaic SCN2A variant? So typically most genetic testing reports will have a table with the gene, the variant, and then there will also be a category for zygosity. And if your child were to have a mosaic SCN2A variant, so again, if only some of the cells were to have that variant, this should be noted on your genetic testing report. Sometimes the language will be apparent somatic mosaicism. Sometimes you might just see the word mosaic. And again, this would indicate that only some cells have that genetic variant while others do not. Oftentimes, when we have a report that says mosaicism, we'll also have a percentage that goes along with it. And this basically represents the percentage of cells where they saw that genetic change. However, something to keep in mind of mosaicism is that, again, on a genetic testing report, that percentage is based off the sample that was submitted. So if we send in a saliva sample and it says 13% mosaicism, we're only talking about saliva sample. It is very likely that the percentage of cells affected, let's say in the brain, is very different. And there really is no way for us to know what the percentage is in other tissues of the body unless we tested all of them, which is, often not feasible and might not be, anything that would change prognosis or diagnoses. And if your child had a SCN2A variant that was present in all cells of the body, then that zygosity would say heterozygous instead of mosaic. Again, if an individual with is found to have mosaic SCN2A, there would be up to 50% chance of passing on the variant if they were to have children in the future. So it's similar. So if someone is heterozygous, there is 50% chance. If they're mosaic, there is that up to 50% chance. And again, there's no way for us to know what percent of sperm or egg cells are affected with that variant. However, just to reiterate, if a child is found to have mosaic SCN2A, the risk for those parents to have another affected child would be close to 0. And again, that's because that change would have happened for the first time and the individual was not present in either the egg or the sperm. However, something I wanted to highlight is that there have been reports of what I alluded to earlier in the presentation, which is germline or gonadomosaicism. And what this means is that a person is, typically unaffected. They do not have any symptoms. But we know that maybe just the sperm or just the egg cells contain a genetic variant. So then there is a risk of not passing that on to children. Typically, when we think of germline or gonadomosaicism, it's often something that's incidentally discovered. Whenever, let's say, a family had an affected child, they tested positive for SCN2A. And now we test parents, let's say either through blood or saliva, and both parents come back negative on the genetic testing report. So we're now living under this assumption that this was likely de novo, very unlikely to recur again. But then let's say that family now has another child and they are again affected with SCN2A with the same variant as the previous child. This would be very high suspicion for us that one of the parents has gonadomosaicism. So again, in that case, we'd expect the parent to be unaffected, not presenting with any symptoms, saliva or blood came back negative. But again, in that case, because they have multiple affected children, we would expect that either egg or sperm cells have that variant. And again, this is usually incidentally discovered because we are not, routinely evaluating sperm or egg cells for genetic variants. And I just wanted to close with this is, a lot of you are probably aware of this and, mentioned in the previous presentation that there are artificial reproductive technology or ART methods to reduce the chance of passing on the SCN2A variant to future offspring. And this would be through a method of in vitro fertilization or IVF, where you could do something called PGTM or pre-implantation genetic testing. And in this case, you could specifically test embryos for that SCN2A variant that was found in the family. And in that case, you could then select certain embryos for implantation based on the test results. And that's all I have for you today. Thank you.